All right. Hi, everyone. Welcome to today's SLAM seminar, which will be given by Thomas Vidabæk, who works in the Rogers Lab at Brandeis University. Thomas got his undergraduate degree at Stony Brook and completed his PhD at the University of Chicago with Sid Nagel. And he'll be telling us about some new routes to self-limiting self-assembly processes.
Cool. Thanks so much for the introduction, Dan. And just if is it okay if people ask questions during the talk? Okay, cool. So yeah, I encourage you if you have questions that come up, you don't have to wait for the end. I'm happy to answer in the middle of things. But yeah, today I want to tell you about some fun and interesting experiments we've been doing at Brandeis University, looking into this kind of class of self-assembly system called self-limited assembly. And just to get everybody on the same page. Oh, are my slides progressing? There we go. To get everybody on the same page, what is self-assembly? It's the idea that if you have some system of particles that have local interactions, their set of local interactions lead to some emergent structure. So you can think about this as like if you have a set of puzzle pieces and somehow they can diffuse around and bump into each other, if they have cohesive interactions, they can form structures. And in this example of this kind of puzzle piece analogy, you can imagine that there are both cohesive bonds and let me see if I can get this laser pointer. There's both cohesive bonds where you have an energy benefit for binding, but you also have a penalty for having unsatisfied bonds. And so if you have this simple kind of square system, you can imagine that as your assembly size increases, you get less and less unfavorable surface area, and you get more and more cohesive bulk. And so these types of particles will tend to aggregate and form large structures. And this is something that's very commonly found in nature. In particular, you can think about crystals that grow from atomic scale building blocks. And even though you have interactions on the smallest of scales, right, between atoms, you can get these really large scale structures. So here, I love this image. It's from one of these crystal caves in Mexico, where naturally these very long crystalline structures can form. And for scale, there's these little orange people here, right? These are meter-sized humans. And so really you can get enormously large length scales from self-assembly. And also it's something that's very simple to do. You may have even done it as a kid, making something like rock candy, right? And so one question that kind of emerges is like, why is this a fun type of system to look at experimentally or in the soft matter field? And one of the reasons is that if you have these kind of natural self-assembly systems on these very small length scales, it's hard to ask questions about the kind of dynamics of these systems or the early time aspects of self-assembly. And so in soft matter, people have looked towards making model systems that can investigate this type of behavior. And colloids have really come forward as kind of a model system for looking at this. And so I just want to show you one example where we have these kind of small beads suspended in solution, and we have some depletants in there as well. And the depletants act as a driving force to prefer these particles to be close to each other. And so in this example here, you can see very rich dynamics of particles coming together, dissociating, and eventually forming larger and larger structures. And colloids are nice because they're typically on, hundreds of nanometers to micron scale, and so they're really well suited for optical investigation. But another nice aspect of them is that you can really tailor their properties independent of their kind of size, right? And so what this means is you can play around with their interparticle interactions or their shape. Here are two examples. This one in the middle is a system from the Rogers lab where you take polystyrene beads and you coat them with DNA. The DNA on the particles can interact, and you can form these nice FCC type lattices here. And you can also see them under optical and confocal microscopy. Or this example on the right here is from David Pine's lab at NYU, where they make these really nice tetrahedral particles that interact from their patches at the center here, and they can make these nice well-ordered diamond lattices. And so this type of view of taking these kinds of chemically synthesizable building blocks and then looking at self-assembly has been very fruitful. But in the Rogers lab, we've been interested in looking at examples of self-assembly in biology. And so here's a nice example of biologically self-assembled structures from the protein database. And we can see that a lot of these are not crystalline objects, right? They have very complicated shapes. They're typically limited in size. And I want to pull out three examples here of kind of microtubules, viral capsids, and the ribosome, where these things have well-defined length scales, like the diameter of the microtubule or the size of the capsid. And so these types of things are not really that well captured by these colloidal systems, right? And so why is this interesting? Why are these length scales interesting? And it's because typically they're related to some kind of function in a material. And so we have three examples here. There's on the left a viral capsid. In the middle, there's this kind of chitinous network that forms in butterfly wings. And on the right is this network of fibrin. And all three of these have a well-defined length that is related to their function. The viral capsid, the diameter, is important for determining the size of the void on its interior, which is necessary for packaging its genome. In the butterfly wing, the spacing in this network is related to its structural coloration and its iridescence. And in the right, though it's a little bit trickier to see, these fibrin networks have a well-defined diameter that's a result of some kind of twisting of individual fibril filaments, which we'll talk about later. But in here, the kind of size of these characteristic bundles is related to these network properties. And so we can ask, if these are interesting types of systems that biologists have made, can we make similar types of structures synthetically to hopefully have some other model system for studying this class of self-assembly problems? And if we look at what kind of we're able to do synthetically, there's a lot of room to grow to make these types of structures. So on the left, I'm highlighting here examples of these types of filamentous or cylindrical shells, kind of analogous to microtubules. And you could say, what can we do with kind of colloidal particles? And so here are examples of kind of lock and key colloids or Janus colloids. And you can make filaments, but these are very different from microtubules, and then they're not hollow, right? Making these kinds of hollow structures is challenging with spherical particles. And this example here on the right are classes of fullerene or carbon nanotube type materials where chemists are able to make spheres and tubes, but there's a lack of control over the properties of carbon. And so you kind of get what you get. And it's really a question of how do you do your synthesis, right? And so here are some examples of carbon nanotubes where in the same solution, you can get different kinds of multi-walled structures, or you can make these more complicated kind of helicoid or helical tubule type structures, but you get a very wide variety of morphologies from the same building blocks. And so what we've been interested in is trying to think about, can you make a good synthetic analog to the colloid, but for these types of self-limited structures. And so to think about this, we want to talk about what are the different pathways that one might take to try and achieve self-limitation, right? And so we can look at these biological examples we highlighted before and ask, how does nature achieve these types of self-limited structures? And so in the self-closing case, on the left, we have things like viral capsid and microtubules. These are examples of assembly where you have individual building blocks that are typically very similar to each other. In the example of many protein capsids, you actually just have one type of protein subunit. But when these proteins come together, they typically will bind at some predetermined angle. And so they can accumulate curvature as they come together, and they form these nice closed membranes, right? And it's the fact that you're accumulating a well-defined curvature and that this thing can close on itself that gives you your size. In the middle, you have other types of examples like the ribosome, where you just have a bunch of unique subunits. So you have a bunch of proteins and RNA, and each one is different and has a unique kind of address in the final structure. And so if you have, it's kind of like an actual jigsaw where you're making a picture, each one is unique and has a well-specified home. And so your yield might not be great. You might have to do extra work to drive it towards that state. But that's also a way of making a finite sized object. And then the last example here of this kind of fibril bundle is another class of assembly called frustrated assembly, where people think these fibril bundles, they twist around each other. And as they twist, they can accumulate internal stress. And they think that this internal stress accumulation leads to the limitation of the diameter of these things. And so what we're going to talk about today is kind of what are the ways we can incorporate these three forms of self-assembly in synthetic systems? And so let's start by talking about this kind of self-closing mechanism. And so we can think about, what are the ingredients we would have to incorporate into a particle to try and achieve self-limitation through this self-closing, right? One of the things is that you want to have a programmable binding angle. So you want to make sure that you get the right type of curvature in your assembly. The other thing is that you need to have something that we call valence control, which means that if, as opposed to these spherical colloidal particles, where they can kind of interact anywhere on their surface, you want to have specific locations of interaction. Because you don't want to have some well-defined angle, but then if you bind to any part of your particle, that's not a great thing to do. And you also want to have some sense of interaction specificity, where if you have different local symmetries in your shape, maybe you want some particles to bind to other particles. And so these are the types of things we can do or we want to do. And if we ask, how can you do this? You might think, well, you have to have some kind of way of tuning the geometry of your shape, right? In proteins, individual protein subunits have a wide variety of complex geometries. And so maybe that's the route we can take to make our kind of synthetic colloidal system. And so what we're going to be talking about for the majority of the talk today is doing that via triangles. So a triangle is a nice particle because it has well-defined edges. Here we can kind of see on the side that there's some little tilting going on here. So maybe you can do some beveling of these structures to achieve angles. And we'll see how we can try and incorporate this in an experiment. Now, it's nice to think about things abstractly, but in self-assembly, it's good to be concrete in talking about what types of structures you actually want to make. And so for this example, what we're going to be thinking about is how do we take and design triangles to try and make these types of cylindrical hollow tubules, kind of an analog to a microtubule. And so one way to think about how you could make a cylinder is you could imagine taking a piece of paper, a flat plane. Here we're going to discretize it just by making a triangulation on it, and we're going to fold it onto itself. And the process of folding a piece of paper just means that you define 2 points on this tiling that you want to be periodic with each other. And for a cylinder, this also means you have this kind of line of periodicity where any pair of points that lie across this thing want to be periodic. And so what that might look like is here we have a set of triangles in a lattice, and we have these 2 vertices here colored, and we're going to pinch those together to make a cylinder. And so now you can, and so that's how you take one of these kind of triangulated tilings and fold it in on itself to make a 3D closed structure. And the nice thing about this structure now is that you can look at it and you can think about what angles you might need to place into your particle along these 3 different lattice directions. Now, before we get too far, I just want to introduce a little bit of variable definitions. So here we need to be able to identify what this discrete tubule is. And so we have this pair of numbers here, m and n, which are just 2 of these triangular lattice directions. And these 2 numbers are going to denote, a pair of these numbers is going to denote how you walk from one of these closure points to the other. So here you just take m steps in one direction and n in another. And so for this example here, this was a 10-4 tubule. And you might ask, why do we care about tubules? And it actually turns out that tubules are a really nice class of structure for this type of problem, because just by changing the angles on these 3 different sides, you could theoretically access whatever circumference or whatever helicity structure you want. And you can do it with just a single type of subunit. So it's a really economical system. It's economical in the sense that you don't need to make a lot of distinct components, and it's super variable. And so what we're going to do is we're going to pick a single type of tubule to start with. So here we're going to think about a 10-0 tubule. This just means that we have no helicity for this kind of white lattice direction, and we have 10 subunits around the circumference. And so this question of, what are the actual numbers you need to program into a particle This was something that was figured out by Douglas Hall at UMass Amherst and Daichi Hayakawa, who was a graduate student here at Brandeis. And so we can then ask, what are the particular properties we have to imbue into one of these triangular building blocks? And the first thing is you want to get the kind of curvature of this tubule. And so you need specific binding angles on each of the three sides of this particle. And so since this thing is kind of symmetric, the angles on this red and blue side are both the same. They're 20.8. And on this white side, you actually need to have a negative angle, just an opposite angle, so that you don't try and make a sphere type shape. You also need to have the ability to control individual sides so that they're not the same, right? And you have to have some kind of binding between sides. And we're going to end up doing that with single-stranded DNA. But you could also imagine doing this with kind of lock and key type structures. And so if we think about all these requirements for this kind of triangular building block, this is very difficult to achieve from just a spherical polystyrene colloid, right? And so we need a different type of technology for achieving this. And thankfully, there's a really nice nanotechnology that's been developed for maybe, I think it's 20 years this year since the kind of inaugural paper came out. But there's this cool technique called DNA origami. And the idea of DNA origami is it uses the fact that DNA is very sequence specific and it's fairly rigid. So if we think about a double strand of DNA, right, it approximately forms a cylinder. And the nice thing about double-stranded DNA is that when it's hybridized, it actually has quite a long persistence length. It's about 50 nanometers. And that's in comparison to the average length of a base pair, which is about in this kind of in this axial direction is about a third of a nanometer. And the nice thing is that DNA is also sequence specific. And so in case it's been a while since you've thought about DNA, sequence specificity means that you have this set of 4 bases, G, C, T, and A, and T wants to bind to A, and G wants to bind to C. And so if you have 2 complementary strands of DNA, which are denoted here by these black and red arrows, and they have the right sequence, these things will very specifically bind to each other in solution. And what DNA origami does is it uses the fact that there's this very specific sequence binding to create 3D structures. And you can think about this as if I had a very long strand of DNA and a very short strand of DNA, and these 2 parts have complementary regions, you can actually pinch this very long strand if you have it complementary to the tails. And so this idea of having short sequences of DNA that bind like non-local regions of another strand of DNA lets you take a very large loop of single-stranded DNA. Typically, this is a plasmid that you've purified from a virus or something like that. And then you can make these kind of local pinches. And if you were clever about how you design all of these pinches, you can fold this loop into things like a smiley face. So this is one of the first things that was made back in 2006. And people have really gone on to make far more intricate structures, 3-dimensional structures using this. There's a cool example where someone made a tiny nano robot where they could actually have the arms actuate, or you can make these kind of larger enclosed shells. And so this is the technique that we're going to use to make our triangular subunits. And so just to give you a sense of what this thing is like, it's really crazy to think about, right? So if you have these 8,000 base plasmids, these short sequences you have here, so this blue strand here is this 8,000 base plasmid, and you have these short sequences that are maybe 40 bases long, you're mixing 200 different strands together in solution, and somehow these things are cooperatively binding to give a well-defined 3D structure. And so this is a nice movie done by the Dietz lab just to emphasize this kind of cooperativity of binding and how these strands bind disparate regions to form these very compact objects. And just as a disclaimer, I will say that this is run in reverse. So this simulation was actually, you started with a structure and then you melted it. But I think it illustrates nicely the concept of DNA origami. And so we can use this technology to make our triangular building blocks. And so here is a TEM image of some of our monomers. You can see these nice little triangles here. They're about 50 nanometers in edge length. It's hard to see in detail exactly what the particles look like here. And so we typically, when we make origami structures, we use a technique called cryogenic electron microscopy, which is a nice tool we borrow from the structural biologists. And you can actually get three-dimensional reconstructions of very small objects using this. And so this is a cryo-EM map of one of our particles, and you can really see some nice structural features. One of them, can I go back to a pointer? I'll just play it again. You can kind of see on the sides of this object, there's a little bit of a tilt on each face. And you can also do these kinds of cryogenic reconstructions on pairs of particles. And so this is a pair of particles, and you can see a slight angle between these two things. So these dimers are forming well enough defined angles that you can get a reconstruction of it. And if you enhance the kind of resolution of this technique, this isn't just a void. There's actually small strands of single-stranded DNA that are holding these two things together. And so,
Should I think of this tilt as a
Mhm.
twisting from front to back, the way we're looking at it on the left?
Yeah, so this is an error in design, this twisting. You have a good eye, Dan. But the way that we envision designing this thing is you kind of make a bundle of helices, and then you just twist them about some center line. And so they should, yeah, it's not necessarily by taking things and twisting them, but it's kind of a rotation of a bunch of helices. And by playing around with the relative lengths, you can control this angle. Does that make sense?
I think so.
Okay. Yeah, it's a little technical. I'm happy to talk about it more later if you want. But we're able to make these things, and we can also tune, importantly, you can tune the angles of these different sides. And so now that we have these subunits, we want to see, can they assemble into these types of tubule shapes? And so since we're mediating our interactions with single-stranded DNA, we can play around with kind of two knobs to change how strongly they'll bind to each other. The first one is the amount of salt you add in your system. This is a very weak knob, but you do need salt because DNA is intrinsically negative, and so you need to screen out any electrostatic repulsion between these things. And the other thing you can do is play around with the temperature. So DNA hybridization is fairly sensitive to the temperature, and there's a well-defined kind of melting transition for any complementary strand, where if you're at temperatures above this melting transition, they won't bind, and below it, they'll basically stay bound for a very long time. And so what we see in experiment is that at high temperatures, here it's around 38 degrees Celsius, we just have a bunch of monomers on our TEM grids. At low temperatures, 32 degrees, we form these malformed looking aggregates. You can kind of see that you get some well-defined width here, but there's lots of holes, they're bent. So this isn't great. But then if you tune your temperature just right, you can get to a regime where binding is weak enough that you have appreciable off rates. So things can bind and unbind, but the binding is strong enough that you can eventually make these kinds of well-formed structures. And so this seems great. We've made a nice looking closed tubule here. And we can so we can ask, how well did we do? And if you look around on these TEM grids, you'll actually find that there's a wide variety in the morphologies of tubules that you see. So here are three examples where one thing that pops out to me immediately is that they are clearly different widths, right? And so there's lack of control in the diameter of these things. But the other thing that's a little bit more subtle is that these structures also have different helicities. So if you look very closely at the patterning of these three structures, you can see they look a little bit different. And the reason these don't just look like triangular lattices is that a tubule, when you're looking in TEM, you're really getting a transmission of your object. And so you can imagine that you can have two triangular lattices that are superimposed on each other. And if they have different helicities, these two lattices will be twisted with respect to each other, and they result in these kinds of moire patterns that you see here. And so we can quantify this variety in our assemblies a bit better by looking at a distribution. And so this is a plot of the distribution of tubules that we see. There's this M and N number. You can kind of think of the M number as mostly controlling the width of these structures, and the n number is controlling the helicity. Just as a reminder, these mn numbers are this kind of path, the smallest path around these closed structures. And each one of these points here corresponds to a different assembled state, and the size of the point tells you, how probable is it to form this thing. And so what we see is that even though we tried our best to make a specific angle and a specific structure, we get a wide variety of tubules instead. And as Dan pointed out earlier, there was a little bit of twist in our monomer. And so even though we were targeting this 10-0 state, we get something that's closer to 8-3 or 9-3 as the most preferred structure. And so why is it that this assembles such a broad distribution of states? This is actually a very ubiquitous problem in these types of self-closing assemblies. And the reason for this is that these subunits are small and they're typically quite soft. And so even though you try and design a monomer to have a specific assembly pathway, so here you just have these kind of little cartoons of wedge blocks, and if they have the right angle, you can make it so that they perfectly would form a ring of 10. But because of their kind of softness, they're very susceptible to thermal fluctuations. And so this means that you can fluctuate as you're growing your ring. You might close early, or you might stay a little more open and incorporate an additional monomer. And so this kind of cooperative fluctuation of many bonds can lead to a distribution of these closure sizes. And this simple view from this ring example carries over quite nicely to the example of these tubules. You can kind of think of these triangulated objects. When they first start out, they start to make a disk, and this disk is slightly curved. And at some point, this curved disk is going to fluctuate and close on itself. And maybe it closes a smaller diameter or a larger diameter, or maybe it closes a slightly twisted way. And so we can try and ask about, what are the expected fluctuations you would expect to see on these types of growing membranes. And so if you think about the elastic energy of a sheet as kind of being spring-like harmonic, so you can say the elastic energy is related to the rigidity of your sheet times its area times its deviation from its preferred curvature. And if you ask what happens when this elastic energy is comparable to kT, this tells you that around the point where this sheet can it's large enough to close on itself, it tells you that you should expect fluctuations in their circumference to scale like the size of the preferred circumference, and it's related then to the bending rigidity. And so you can take this type of energy and say, oh, if my system is growing slowly enough, probably these different closure states are Boltzmann distributed. And so you would expect some kind of Gaussian distribution of these states. And that's somewhat similar to what we see in experiment. And you can actually take this type of very simple harmonic energy model and ask, what is the fluctuations of this distribution? And you can pull out some number for the expected modulus of the system, and you get a number that's around 7 kT. And this is fairly weak. I think people think that lipid membranes have a rigidity of kind of 20 or so kT, so this is quite a floppy system, right? But we actually recently have done a little bit better than just using this type of estimate. You can actually use cryo-EM to make more quantitative measurements of the rigidity of your particles. This is an image of cryo-EM, so this is a little different than in TEM. This is a system where you can trap particles in a very thin layer of ice, and so you can get access to different orientations of them and different kind of instantaneous conformations of these particles. And so by preparing samples of dimers, you can capture dimers in different configurations within the ice. And so you can get not just the kind of average structure of your dimer, but you also have access to kind of the range of states that they can accommodate. And by making these types of measurements, you can see what are the different deformations that these pairs of particles can allow. So we find that aside from just having bending deformations, these particles can also twist a little bit when they're bound. And so we've done some work where you can do a very quantitative look at these types of deformations. For instance, you could look at distributions of binding angles for the different sides of your triangle. So here, remember for the 10-0 case, we had these angles of 20, 20, and -10. So this is where this kind of twist story came in, right? We had 18, 9, and -4. And so these kind of wrong average angles are why our state wasn't a 10-0 tubule. But you can take these types of, of measurements and ask, can you make predictions about this type of distribution of states that come out? And you can make some fairly nice predictions. You get pretty close just by considering these types of pairwise interactions between particles. Okay, and so this is kind of this first arc of self-closing, right? We're able to make these types of, we have this kind of new playground of synthetic subunits, similar to colloids, but now we can make these types of self-closing structures. And we ran into this problem where thermal fluctuations give rise to this polymorphism, right? And we can kind of measure it and explain it, but it makes it tricky, right? If you wanted to use these types of self-closing things to make well-defined structures, you're kind of always butting your head against the fact that you'll probably make a distribution of states. And in natural systems, biology finds typically other ways to avoid this type of polymorphism. For example, in microtubules, they have this thing called the gamma tubulin complex, which acts as a seed that helps nucleate the right type of structure. There's also kind of assisting proteins that bind to it and help it stay on target. And so we want to think, is there other ways that synthetically we can try and get rid of this polymorphism? And that takes us to the second part of the story, which is to think about how this idea of addressable assembly can be used to help limit the polymorphism that we see in this class of self-closing structures, right? And so there's this problem, polymorphism, and what we're going to do is we're going to use addressability. And so if we think about this example of this ring type assembly, right, where we had these polymorphic states, we could think, maybe if we had two particles instead of one, right, we had a red and a blue, and red only binds blue and not itself. This would naturally disallow these odd numbered states, right? Because here you'd have to have red binding with red or blue binding with blue. And so naturally, by introducing a little bit more complexity, we should be able to remove off-target states from even being allowed to form. And we can also do this in this example of tubules. And so here are two neighboring states. There's a 10-0 and a 10-1. And what we can do is we can take a fictitious paintbrush and we can think, okay, what if I had stripes of blue and stripes of red particles, right? And so we try and design these types of interactions where you only have red binding in this kind of lateral direction and red-blue binding in this axial direction. And what we see is that if you try and make this 10-1 tubule in the same type of striped patterning, you would get this seam of unintended binding. And so for this example of kind of striped assembly, if we ask in this kind of MN space, what are the allowed or disallowed states for assembly, we find that half of the states get removed, right? So all these odd numbered end states get thrown out. And so in general, this idea of adding in complexity to make more and more complicated assemblies is a way of pruning the density of states of your assembly. And so,
what should we make of the fact that nature chose the one with the seam for the microtubule, the most common kind?
oh,
I don't know if that's a fair question since you're not
yeah,
aiming to replicate it exactly.
yeah,
But
I'm not sure. I think I don't know enough about microtubules, but there's probably the reason it's chiral is probably related to some function. Maybe it makes walking easier, or it has to do with like how it depolymerizes, but I'm not sure.
okay.
Yeah. Um, yeah. Okay. And so this idea of adding more complexity seems appealing. And so how can we achieve different types of colors in our experimental system? So say we wanted to make this 3-colored tubule here. Well, the nice thing is that since our monomer just has single-stranded DNA coming out of the sides, we can leave the base geometry alone, and we can just change the sequence on the ends of these different sides. And so you can imagine taking if you have 3 different particles you want in this system, you would just individually fold the kind of core of these particles, and you would swap out the strands on the sides. And if you design the sequences of these extruded strands just right, you can introduce whatever kind of interaction scheme you want. So in the case of this tubule on the left, you would have these types of interactions where you have some self-complementary side on each particle, and then the other edges are kind of interparticle interactions. And so now the question becomes, okay, we have a way of introducing complexity through this kind of tiling of colors into our particles, and we want to know how much complexity is enough, right? It's this kind of continuous variable where you can go from having just a single particle species up to, you color every single particle on your structure. When do you stop? And so what we did is we're experimentalists, so we just kind of ran at it. So what we're going to do is we're going to just kind of crank up this complexity knob. So we start by assembling tubules with just this one color. This is this distribution that we saw before. And then we can start increasing the complexity. So we go to a 4-color scheme and we see 2 features pop out in this distribution. One is that we actually get really good isolation of these off-target states. This coloring should disallow both odd M and N numbers. So we see that here. And we also find that the most probable state has a higher probability than in a single color system. And if you keep doing this, you go up to 16 components in your assembly. We really get a majority of the assembly in just a single state. And so here we go from having kind of the majority states being around 13% in one state for the one component up to 94%, almost full yield in the 16 component system. And you might be worried that these 16 component things are like very tiny or highly defective, but they actually can make quite long structures. And in general, they're actually less defective than the single component thing due to some weirdness that I don't want to talk about today. But you can ask questions about it later. And so this seems like a really nice way of both of increasing the kind of selectivity of your assemblies just by decreasing the density of states. And so what we can do is we can look at what is the yield, the selectivity of your most probable tubule state as a function of the complexity, here the number of colors in your system. And what we find is you get this kind of monotonically increasing dependence on yield until you reach 100% and then it starts to plateau, which is great. But we want to see how does this depend kind of on more general features of your self-limiting assemblies. And so we went back and we designed a new monomer. We learned about this twist error. And so we tried to target a 6-0. And so here's a distribution for a one component kind of smaller diameter tubule. And we hit 7. So we're doing a better job now of getting the state we intend. Just as a reference, this is where the center of the previous distribution was. And again, we can crank up the number of colors in the system. We can go to 4 or 7 components. And we see the same story where you lose access to states and you increase the yield of your system. And if we go back and we again plot the kind of maximal yield as a function of the number of colors for this narrower tubule case, we again see this increase in plateau, except it's happening at a smaller amount of complexity. And so we want to ask, wouldn't it be great if we could scale this data together? And so we want to think about what would do this. And so one way I like to think about these types of tubules is instead of thinking about this closed structure, I think about on this tiling, there's some target state. And this target state corresponds to this closed tubule. Say this is a 12-0. And what we've seen is that there's fluctuations, Gaussian fluctuations around this state. And so there's some area around this target state that's accessible to the system due to these binding fluctuations. And what we're doing is by adding colors, we're essentially changing what states are able to be closed. So if your target state has some local symmetry or some local coloration, only states with similar local vertices are allowed. And so by increasing your components, you're reducing the number of states in this same area of fluctuations. And you're introducing a length scale lambda, which is the separation between these similar vertices. And once you get to a large enough number of components in your system, you can make this lambda big enough so that your next available state lies outside your range of fluctuations. And so we can use this kind of intuition to get a scaling law. So if we think back to this example of kind of the elastic energy of a membrane, we had that the expected circumferential fluctuations go like the circumference over the square root of the bending rigidity. And so that means that this kind of accessible area goes like c squared over b. This separation lambda roughly goes like the square root of the number of components. And so if you balance these things together, you get that the optimal number of colors goes like c squared over b. And so if we go back to our yield data, and we take this number of b that we had from earlier, we can rescale our data, and we get this very nice collapse. And you can do some simulation work and some kind of simple theory work to get a predicted curve, and it matches up very nicely. And so,
If I understood your arguments,
yeah,
that would establish n star colors as a threshold minimum, but why is it also the optimum?
I guess it's the optimum in terms of if you're already getting like 100% yield, or you're getting
Hmm.
very close to 100% yield, there's no more benefit of increasing your complexity from there. One obvious trade-off is that the timescales for your assembly are going to drop. So if you're in a diffusive regime, the more and more different components you have in your system, it's going to make kind of your on rates fall by 1 over the number of components, and so that's not good. And
Hmm.
we actually saw for these narrow tubes, we went up to kind of we had almost full selectivity at 7. We went up to 14, and we started to see other failure mechanisms show up. So there also seemed to be a downside to being too complex. And so that's why we think there's this kind of optimum assembly complexity. But one thing as an experimentalist that I find a little scary is that this thing scales with c squared, right? So if you're trying to make well-defined tubes and you go to bigger and bigger diameters, this is really going to kill you in terms of, how many components you have to put in. But one thing we noticed was that you can actually get slightly improved scaling if you're willing to have some trade-offs. And so we considered this type of linear tiling here, and we noticed that these linear tilings are actually able to constrain the width of these types of assemblies. And so here is an example of one of these tilings where these red lines here are just something where we've decorated gold nanoparticles along these red particles. And if we look at the polymorphic states from this type of linear tiling, we find that all of these states basically just have the same width, which is really cool that you can do that with the system. And if we look at how well we can control the width of tubules, then as a function of the number of colors, we find that we get collapse of our experimental data with a theory curve. Now, instead of being 1 over c, it's 1 over c squared, it's 1 over c. And so this is nice. So if you're willing to trade off on the fact that now you have fluctuations in your helicity in your final state, you can really well constrain the width of your structure. And so this is a nice workaround. Okay. And so I've kind of told you about these complex structures we can make. We have control over angles. We have control over specificity and interactions between particles. And recently, what we've been working on is trying to make more and more complicated types of geometries than just tubes. And so one example of a complicated geometry is this example here of a triply periodic minimal surface, or Schwartz piece surface. And our friends at UMass Amherst found a way to do subtriangulation on these types of more complicated negative Gaussian curvature surfaces. And so recently, a graduate student in the Rogers lab has been working on making these an experiment. And so we're able to get these kinds of porous structures, which are similar to this kind of butterfly network we saw earlier. And there was some really cool work by Mason Price, an undergraduate in our lab, who was thinking about what if you take these types of tubule structures and you do some cutting and stitching to try and make these toroids or helical structures. And he was also able to realize these in an experiment using this type of platform. And so we've been really excited about the kind of doors that this has been opening for this type of self-limited assembly. And so what we saw in this kind of middle part is that addressability is really good at controlling the kind of selectivity of your structures, and then it's also good at controlling the kind of giving you access to these more complicated geometries. And in the last part, I wanted to talk a little bit about some preliminary work we have on trying to access this last case of frustrated assembly, though I see I'm running short on time. So Dan, just let me know when to wrap up and I can speed through it. Um, and so frustrated assembly is intrinsically quite different than this idea of self-closure. So we can consider two particles here. One of them is just a square. So it's kind of what we saw in the beginning of the talk. And then you can imagine taking a square and on the sides here, you just tilt the angles in ever so slightly. And you can ask, how do these things fit together? And so in the case of the square, you can tile the plane very nicely. But when you start to take these kind of tilted particles, in order for these things to bind to each other, you actually have to deform their edges a little bit, right? This shape won't tile the plane. And so if you allow for elastic deformations of your particle, you can force it to tile at the expense of accumulating some kind of internal stresses. And so in the example of the unfrustrated case, you have a free energy curve where there's kind of there's a balance between surface cost and bulk gain, which gives you kind of a classic nucleation and growth looking free energy curve where if you can make a large enough nucleus, you can then access unlimited cluster sizes. But in the case of frustration, typically this kind of deformation of a particle leads to a super extensive elastic frustration, which if you add it to the surface cost and bulk gain gives you a curve that looks like this for your frustrated assembly, and it leads to some preferred size where you're balancing between the benefit of binding and the penalty of deforming. And so we've been working with Kyle Sullivan at UMass Amherst on kind of an ideal version of frustrated, or a simple version of frustrated assembly where we consider these things called curvamers. So Kyle has been thinking about these little curved platelets that have a well-defined radius of curvature, and they have some adhesive interaction top and bottom. So these plates want to try and stack on top of each other. But because of their radius of curvature, if you take two identical plates and you try and stack them, you're naturally going to form a little gap here. And so there's two regimes that these particles can live in. One of them is that this attractive interaction is broad enough so that when these particles come together, they actually don't care that there's a gap and they're happy to just stack on top of each other. This would lead to kind of unlimited assembly where you just get a filament that can keep adding stuff to the top and the bottom. The other regime is that if you make this attractive interaction very local, very narrow, then these particles, in order to satisfy this bond, would have to deform, right? So these two particles, if they have conformal contact all along their surface, they would have to have different radius of curvature. And this is exactly what we need, this kind of super extensive deformation to lead to self-limitation. And so we've been trying to implement this in DNA origami. And the way we think about this type of design is pretty different from the design before. For the idea of the triangles, we basically just thought about DNA as being rigid blocks, and you use that to build up your shape. But here, what we're going to think about is just having an origami particle that's essentially two different layers. And if you can tune the inherent length of each layer, you can get this thing, and you enforce kind of boundary conditions at the end of the structure, you can force this thing to bend, kind of like a bimetallic strip. And so we're able to do that. This is a TEM image of monomers of this particle. And so we can see that there's lots of little curved platelets here. And we can actually go in and measure what's the radius of curvature of different monomers, and you get a fairly well-defined distribution. So this is a distribution of the radius of curvature normalized by the length of the monomer, and we see a well-defined peak here. And you can also use the fluctuations here to get some sense of how rigid these things are. So this number, just to give some meaning to it, you can kind of think about this as being the persistent length of these curvamers. And for scale, the length of a curvamer, the arc length is about 200 nanometers. And so we can design these little curved disks, and we can incorporate interactions to these things. We can add single-stranded DNA on the top and the bottom. And when these things bind in solution, they form these nice little stacks. And if we zoom in on one of these stacks, we can see these pair of lines These are single curvamer, and they seem to be pretty well stuck together. Dan, how am I doing on time? I don't want to.
So I think 8 more minutes would be the absolute max.
Okay, well, yeah, I don't have too much to say about this, so I'll just mention that one of the nice things about these types of curvamers is if we go in and we measure their kind of the distribution of sizes that we see here, there's two things we might expect. One is kind of unlimited growth, where you would just expect this kind of exponential distribution of sizes. And what we're hoping to see is this kind of self-limitation where you get a peak. And we do see this in experiment. So this is a distribution of the stack height of these types of monomers. And we see a well-defined peak here. We've done a couple checks where, if you change the concentration of your particles, your peak doesn't shift around, which is nice. This means that this probably isn't a kinetic trap. And the other thing we can do is we can try and change the interaction energy between particles. And so here, just by changing the temperature, we can see the distribution of these things changes slightly, right? So this tells you that when you have increasing adhesion, lower temperatures, you can have some variability of the stack height. And along with Kyle, we're now starting to make some comparisons of these kind of experiments to theory. He's been working on some nice MD simulations where when he starts with curved particles, we see that these things tend not to grow larger than 3 or 4 particles for a stack, whereas in the flat case, you can really get kind of much longer filaments. And so we're excited to see where this takes us. And so just to wrap up, hopefully I showed you that this kind of DNA origami platform is a cool way of making synthetic analogs that can mimic these self-closing, self-limited structures from biology. Going forward, we're really excited about this kind of frustrated example, and we want to do some more work quantifying the kind of elastic properties and binding energies of these things and start making meaningful comparisons with theories, see where kind of existing theoretical frameworks are good and where they fail. And going forward, we're starting to become more interested in kind of shape-changing assemblies. It's nice to be able to have static structures, but it'd be cool to have structures that can then deform after they assemble. And so Ryan Hayward had some nice work where he made hydrogels that could deform into different types of shapes. And so we've been thinking about making open lattices of particles where you can pattern their internal stresses to make out-of-plane structures. So before I end, I just want to thank all the collaborators. This work is really a huge effort by the Brandeis MRSEC, and there's many members who are part of the team. I want to shout out the main PIs, Ben Rogers, Greg Grayson, Seth Fraden, Mike Hagan, who lead the kind of experimental and theoretical work. And then most of the work I showed you about triangles is really a lot of work done by Daichi Hayakawa. And the work on curvamers is a collaboration between Morima Roy and Kyle Sullivan. And of course, want to thank the NSF for funding. I'll end here on this summary slide, and I'm happy to take questions. Thank you.
All right, thank you, Thomas, for a wonderful talk. And the floor is now open for questions. Feel free to unmute. Angwoo, go ahead.
Hi, thank you for the great talk, Thomas. I'm wondering so I may have missed this earlier, but it seems like your framework, the final structure is selected at the moment of closure due to thermal fluctuation. I'm wondering in active or driven system, so the system is inherently non-equilibrium. Do you think the proposed mechanism is still hold, or could the structure keep evolving after closure?
Yeah, that's a very good question. I'm not sure for these self-closing systems how much you can get away from the inherent geometry of a particle, But if you think about these kinds of 1D filamentous systems, right? Like if you think about actin, right? Or these curvamers, for instance, you could imagine there being a feedback loop where maybe as they're growing, they are a natural way of forming like inhibitors, you know? So you could imagine having a system where, let's say you're making a stack, and then locally that lets you build up something that feeds back and lets it fall apart, right? Lets it depolymerize. I don't know if I think that might be related to what happens in polymerizing systems in biology, but I'm not entirely sure, right? I know actin has this catastrophe mechanism where if they get too long, they start to depolymerize. But I don't want to. I really don't know anything about that, so I don't want to talk about it. But you could imagine there being systems where you would have to have something that is like size dependent that then triggers it wanting to disassemble, right? But I don't know if you could have that in these self-closing examples. Maybe you could have, yeah, I'm not sure. Maybe if you could have the sides change length, then you could have things where the kind of metric changes internally in the structure, but I'm not too sure. It's interesting though. Yeah, I think non-equilibrium systems are very interesting, but I can't say much about it.
Thank you. Thank you so much.
On that topic, since you just mentioned it would be nice to have actuatable or changeable structures, is there some analog in DNA origami world of protein that changes the conformation of a tubulin dimer in a microtubule? It changes the lattice from less strained to more strained.
Yeah, so one thing you can do, there's like one technique that's like probably good for doing actuation, where in DNA nanotech you can do something called strand displacement. So there's this idea that you can have DNA hybridization, right? But if one of your strands is a little longer than the other one, it has a little tail that's sticking off, you can have another strand that's floating around in solution that's also complementary to this pair, to one of the strands in this pair, and it'll come in and actually kick off the other strand. And so you could imagine trying to make a system where maybe you have some internal struts that are defining angles, and then you can displace these things to have them become more floppy or have them turn on in the first place. And that could be a way of like post-assembly trying to muck around with the kind of internal geometry, which would be cool.
Cool.
Yeah.
Yeah.
I have a quick question about the floppy modes, I guess, that you saw of your triangles, and maybe more generally about how what knobs you have to play around with for tuning your interactions. So I guess all the triangles have these single-stranded DNAs hanging off, which can hybridize with certain other ones. I guess the angles that you see in, for example, in the cryo-EM reconstruction, is that consistent with like the length of the single strands that you have? And could you make those more rigid by having shorter single strands so that they're just long enough to reach and hybridize, or does that have other costs elsewhere?
Yeah. That's a great point. So I think it's fairly consistent with the fact that kind of the strands between our two structures, we have little regions of unpaired DNA. So it's kind of just like little loose polymer, and then a double-stranded thing, which is pretty rigid. And it seems like the size of this single-stranded DNA is what drives a bulk of the kind of floppiness of this system. And so we've done some studies for these kinds of open lattice assemblies where you really drive up the number of these single-stranded domains, and you can change the rigidity by like over a factor of 10. If you want to make it more rigid, one thing you could play around with for these kind of like, if you want to define an angle, is the geometrical placement of where your strands are. So the way that we're doing it right now is on the face, everything's kind of coming out of the middle. And so it's really acting like a hinge. But probably if you can make the face of your origami taller and you have the strands just be further separated vertically, that would also certainly limit how much you can flex. So there's some games you can play, but I think we don't have a good theoretical framework to know what to do.
Mm-hmm.
It's kind of it's expensive to test a lot of these ideas.
Sure.
Right. Any further questions?
Hi, Thomas. I might miss it, but can you describe a little about the process of the self-assembly? Is it, for example, for the tube one, is it grow, or do they start from some small part and then grow on two side by adding the subunit gradually, or they have? Yeah, I kind of want to know the process.
Yeah. It's a good question. So we work at pretty low monomer concentration. And so we're pretty confident that these things just grow from monomer addition, right? There's kind of like two regimes you could imagine. One is that you have a structure and you put a monomer on and you just keep doing that and you take it off sometimes. And we've done some kinetic Monte Carlo simulations along those lines, and they agree fairly well with what we see in experiment. The other option that you could have is that maybe you form clusters of particles and these clusters bind to each other, but I think we're too dilute to be in that limit. But then at some point you run out of background monomers and you have a fixed length distribution. So we find typically for these kind of tubule systems, you get an exponential distribution of lengths of structures. And then the mean of that is just set by your initial starting concentration.
Okay, got it. Thank you.
Okay. If there are no further questions, let's thank Thomas once again. All right.
Cool. Thanks for having me, Dan.
Oh, thank you so much for speaking. I'm going to stop the recording here.